Today I'm going to walk you through the Decoding DNA lab provided by Kansas Corn STEM. All right, my name is Shelly Robinson and I'm a lead teacher for Kansas Corn STEM, and today I'm going to walk you through the Decoding DNA lab provided by Kansas Corn STEM. And if you have previously attended one of our workshops and you have our handouts in our notebook, you'll find this activity in the blue tab section, pages S10 and S through 12. If you haven't come to one of our workshops, I highly encourage you to sign up and get enrolled for one of our summer sessions. The purpose of this lab is to model protein synthesis from the DNA located inside the nucleus of a cell and bring that out through the process of transcription, where DNA is converted to messenger RNA, and bring that out into the cytoplasm where the protein can actually be produced. The outcomes for this lesson are helping students have a better understanding of the modeling of the structure of DNA, helping them identify that the majority of the work taking place in protein synthesis does not happen in the nucleus of the cell, but outside in the cytoplasm. We also want them to understand the two processes of transcription and translation. This unit can be incorporated into every biology class, whether we're in high school or the middle school. Certainly with all of the genetics information that students are exposed to in the media today, they have a growing interest in everything related to DNA, from their own ancestry account or 23andMe, everything that they're hearing about GMOs. There's a great deal of interest in genetics for our students, and so this is a perfect activity for them to participate in. This is a very simple lab to set up. It has low-cost materials. One of the things that we want to have are our DNA sentence strips, and those are provided in the website, or provided on the website. We have both middle school and high school strands, and they're color-coded here just so you can kind of see the difference. You need a set of transfer RNA cards, and I will explain the color differences later. This PowerPoint, which is also located on our website, you can have that. And we have a model of a ribosome, and this is a 3D printed ribosome that you can find the STL file on our website as well. I also like to use a hula hoop, and I think our students recognize the nucleus a little bit easier than saying, this table over here is our nucleus. When I put a hula hoop on there, they get a better visual image of that. So I like to include that and my model of the DNA. The setup is real simple. A lot of it occurs beforehand. Hopefully you have a student aide and they can do a lot of the cutting for you, because not that this is difficult to do, but it's time consuming. So if they can get all of your DNA sentence strips cut out for you and can cut out the transfer RNA cards for you and bag those up, one set per student or for the group of students, however you're going to align your room. So real easy setup, not a lot of expense, which is nice. As science teachers, we probably all have our students working in groups already, and maybe you have assigned groups, but get those students working in a group, have those sentence strips printed out, and then we want to identify some area in our room as the nucleus. I try to make it a central location, and as I said earlier, I try to put this hula hoop out there just to help them know where that is located. One thing you want to remind your students is that these DNA strips cannot leave the nucleus, because in the activity they're going to come to the nucleus and they're going to transcribe DNA into messenger RNA. And they will very often want to just take this back to their table and work with their group, when in fact they can't, because the DNA, being a double-stranded model, is too large to leave the nuclear pores of this membrane. So they have to do the transcription within the nucleus, and that's something that students often they don't try to, but they often will take it back to their table, and we want to prevent that so they have a better understanding of what's taking place and where. All right, as I mentioned earlier, one of the things you needed to have done beforehand is to cut out these transfer RNA cards, and as you can see on the PowerPoint, the cards are color-coded. And if you'll notice, every sheet, the 3-letter codon is beginning with a different letter on that sheet. And so if in the process of translation, your students are looking for a codon that begins with a C, they recognize that all of the C's are in yellow. All of the codons that start with the letter C are in yellow. So it just speeds up their searching process. So I think that's a really nice idea to color code those. This is just a sample setup of what your room layout might look like. Again, central location for that nucleus. So let's talk about how we're going to teach this lesson to these students. So first of all, one of the things that I like to do is to show them some introductory videos. And I'm not going to show these videos today in the interest of time, but I've given you links to 3 different videos that I think are very good introductions for our students. The first one is Mr. Anderson, and everybody knows him. He does a really nice job of comparing DNA to protein synthesis, comparing that to cooking. And specifically, he talks about cooking pizza. But it's a very good analogy that I think our kids can really relate to, whether it's high school or middle school. The other 2 links are to Howard Hughes Medical Institute animated videos. And they're kind of quirky, but I think it gives our students a visual image of what's happening at a cellular level with our DNA, transferring it into that protein. So I think these are great starter videos. So our kids know, they come to us with this lesson already, that they have a good understanding of what DNA is. And they know that it carries our genetic information, and that it's made up of 4 different bases: adenine, thymine, guanine, and cytosine. And it's arranged in this double helix shape caused by the chemical bondings between these different molecules. When we look at this DNA, we need to remember the size of this molecule. And while it's minuscule, it's still too large to leave the pores of our nuclear envelope. The first step in getting our DNA to produce that protein that we want is transcription. And transcription is the process of simply converting DNA to messenger RNA. Remembering that this cannot leave the cell, we have to put this into a different form. And the way that occurs is that we have an enzyme known as Rna Polymerase. And that Rna Polymerase reads the DNA, and it selects one of the 2 strands, and it's going to convert that into messenger RNA. Now, messenger RNA and DNA have 2 major differences. First of all, our DNA is double-stranded. Messenger RNA is single-stranded. The second difference is that our DNA uses the base thymine. Messenger RNA does not use thymine, but anytime you would want to put a thymine, we're going to use the base uracil. So you can see from looking at the green strip of messenger RNA on the PowerPoint that the uracil is replacing every time that adenine on the top row, the top yellow row, would want to replace with thymine, we're replacing with uracil. And so you can take a look at that and see that in that bottom strand there are no thymine molecules. Once our messenger RNA has been created, that messenger RNA is now small enough that it can literally travel through these pores of our nuclear envelope. Now that I like to tell my students that our classroom is the cytoplasm. So now that our messenger RNA has left our envelope, our nuclear envelope, and it's out here in our cytoplasm, we now need another element to help us complete this process to protein synthesis. So we're going to now translate it into a different form. And translation occurs by reading our messenger RNA 3 bases at a time. Those 3 bases are referred to as a codon. In order to begin reading this recipe, we're looking for a starting point. And that starting point is going to be identified by the letters AUG. That's the codon that represents, let's start here. If you can see, and I don't know how well you can see on this particular strip, but a lot of times our DNA has a lot of information before we ever get started reading for the particular code. It's bases that really are irrelevant. And so we're looking for a sign as to where to start, and that sign is the AUG's, excuse me, the AUG codon. Once you have identified the AUG codon, you can now begin reading the other letters 3 digits at a time to find the other codons that you're going to be pairing with. In the cytoplasm of this cell, there are many, many different transfer RNA molecules. And those transfer RNA molecules, as you see in this picture, the transfer RNA molecules have an anticodon. The anticodon is going to pair with the codon that is on the messenger RNA. On the opposite end of that transfer RNA, there's an amino acid. And those amino acids are going to be linked together to build our protein. So in this picture on the slide, you see this green dome, and that's going to represent our transfer RNA molecule. Excuse me. That's going to represent our ribosome. That ribosome is going to bring in those transfer RNA molecules based on the message on our messenger RNA, based on those 3 codes. As it brings them in, it's going to link them to the previous amino acid that was brought in. To start this process, we start with initiation, or the beginning. And we're looking for that AUG codon that is going to be matched with this corresponding anticodon of UAC. So the A is going to pair with the U will pair with the A, and the G with the C. That gives us our starting point, so we can now begin reading. There may have been a whole lot of letters before that, but our ribosome isn't reading the protein recipe until it gets to the AUG. Now it's going to add the next codon based on, the next anticodon based on the CAU. And it's going to bring that in, create a bond between the previous amino acid, and release the first transfer RNA that was brought in. This process is known as elongation because we're taking one amino acid and adding it to the next one and the next one. Obviously, our chain is going to be getting longer. So we continue this process until we've added all of the amino acids to build the protein that our messenger RNA is carrying the recipe for. And we read that until we reach a stopping point. And that stopping point is called a stop codon. And that is typically AUC. We now have termination. We've reached the end of the road, reached the end of the recipe. This is the protein that our DNA originally outlined in the nucleus of our cell, that our messenger RNA brought out into the cytoplasm for our ribosomes and our transfer RNA to build our protein. So if we're ready to start with our students, we would want to give them the handout you see here on the screen. These can be found in the student section, pages 10 and 11. And the students are going to need this. They're going to need their packet of transfer RNA cards. And then they're going to come one at a time from their group. They will come to the nucleus, wherever you have that located in your room. Now, one of the things that you may notice on the 2 strips I've used on the screen, these 2 code for the identical same protein. The strip on the top is more geared towards the high school students. The strip on the bottom is geared towards our middle school students. If you look at those, at first sight, they may not look much difference, but you'll notice the one on the bottom, the letters are already grouped in groups of 3, and it's not nearly as long. So what we've done is we have taken the high school segment, and the part between those red lines is the same as the middle school section below. But our high school students should be able to weed through and look for that start codon a little bit easier than our middle school students. I will tell you, I've taught high school biology for a number of years, and for my lower level classes, I always start with the middle school students' strips. And then I keep some of the high school student strips in my pocket and just kind of flip those in every now and then for groups that are really progressing a lot faster. But I find it easier for my students to start with the middle school and then work up to the high school, because they don't have to weed through that excess DNA that really serves no purpose. So whichever strips you're going to use, whether you're using the high school strips or the middle school strips, students are going to come to the table and they're going to bring with them a blank strip that's just for their purpose of writing. And they're going to select one of the DNA strips that they want to use, and they're all numbered, okay? And when they pick them up, they're going to stay right here. We do not want them to leave the nucleus because DNA is too big to get out, so we want them to stay right here to do their work. And they're going to read this DNA strip, and they're going to convert it to messenger RNA. So we have a DNA strip, and we have a corresponding messenger RNA that they have created using the screen using the slide on the screen. You see that thymine corresponds to adenine. We have an adenine on the top row, which is our DNA gene. Remember, we don't use thymine. We would normally pair that with thymine, but because it's messenger RNA, we're going to replace that with uracil. And so as students go through, they're going to end up with a messenger RNA strip. The messenger RNA strip, they now take back to their chair, back to their group with their table, or with their friends, and they're going to translate that into the protein that we're trying to build. So the first thing they need to do is they need to mark this off in groups of 3. If you're on the high school strips, however, you need to first of all find that codon that says, hey, start here. They're looking for that AUG. Middle school ones, they're going to start with that AUG. Okay? So depending on, who you're working with, will determine what needs to happen next. Once they find the AUG, they now just need to draw a line every 3 codons. They can use the ribosome, the 3D printed ribosome, and thread their messenger RNA strip through here, and they can read those 3 digits at a time. Okay? And that's just a physical model. I think it works really well for, those hands-on kids who may just struggle to do this anyway, because this is hard stuff. And I remind them of that. Once we've identified our start codon with our AUG, now we have to find the corresponding transfer RNA card. So I just happen to pick up blue, but we're looking for the UAC card. And on that UAC card, it says start. And they're going to lay their strip down. And I know this is in the nucleus, but we're really at our table now. We're going to lay our strip down, and we're going to pair it up with our transfer RNA anticodon. So A pairs with U pairs with A, G pairs with C. Then they're going to look for the next one. And I'm going to show you this all on the screen, but they're looking for that next codon, anticodon. So U pairs with A, C pairs with G, A pairs with U. And they're going to continue that process until they have identified all of those codons until they reach the stop sign. Once they've done that, my hand is a little slow today. Once they've done that, now, because we're working with Kansas corn lessons, we have built a protein or a sentence that is Kansas corn related, it's genetics related, it's ethanol related, but the sentence needs to make sense. And if the sentence does not make sense, some genetic mutation has taken place. Now, that mutation may have occurred in the beginning when they transcribed, I'm sorry, too many T words, transcribed their DNA into their messenger RNA. That's typically where the mistake happens. So if their sentence doesn't make sense, I would send the students back to the table, find the same number that they started with. So I transcribed gene 12, so they need to come up here and find their gene 12 again and go through and make sure they have correctly transcribed this into the right code. Okay? One thing you may notice on the screen is that, for example, the yellow card that has the CCG, that card has a number of words that it can represent. Much like our amino acids can be represented in a number of different coding formats, in our lesson, because of the number of words we needed to use, some of our codons and anticodons represent more than one word. And in this case, the kids would have to choose between the words less, decreases, or decreasing to make their sentence flow. So in this case, it wouldn't make sense if the student said ethanol less carbon dioxide production. So they would have to choose the proper word to make it a flowing sentence. So obviously, when they get it completed, they're going to check with you to make sure they've got this process correct. Once they have it correct, they're going to move on, and another student will come to the nucleus, bring their messenger RNA with them to transcribe it into the process that can be, or the format, not the process, the format that can leave the cell to get out and be translated into the protein. You would continue this until all the students had had a chance to go through this process. One thing that I have found is that it typically takes me 2 days to do this particular activity, because the first day they're just trying to get in the flow of it. The second day they can really rock it and get going with it. So I think it's a 2-day lab for me. It may not be for everyone. I've included just an online interactive practice game for the students. It's very game-oriented, but it really does a nice job of going through the process of transcription, translation, and protein. So if kids need something to do for practice, I think this is a really good opportunity. The second part of this activity is basically everything we've just done in the reverse order. So for genetic engineering, which our students are hearing more and more about, how we can manipulate DNA, encourage it to make the proteins that we want to make, whether that's to treat hemophilia or whatever that happens to be. Students can take this process and just do it in the reverse order, and that would be simulating our genetic engineering. So in this case, we would give them the pre-written sentence, and they would take that sentence back to their table, and they would match their anticodon by finding the words that make the sentence. Once they lay out all of those words that make their sentence, they're going to match the anticodon to a codon. So they're going to write the messenger RNA. Once they have their messenger RNA, now they're going to convert that back into DNA. Remember, messenger RNA has those uracil molecules, but not the thymine. Now we're converting messenger RNA into DNA, so we're now going to put that thymine back into the picture. So once students have practiced this a couple times, they kind of get the idea of how we as scientists and scientists of the future definitely are going to be using this process more and more to help cure the diseases that inflict individuals that we come in contact with. One thing I didn't mention is that whenever they're transcribing this, they're going to reverse engineer this, reverse transcribe it. They need to make sure they keep, and I don't have that in my hand, but the start and the stop codons need to be added to their sentence. So make sure we don't forget that. So in conclusion, this is a great activity. It's very low budget, it's very hands-on, and it's a great model of how students can take DNA, transcribe it into messenger RNA, get it out of that cell into the cytoplasm where the real work is done. You know, the nucleus usually gets all the glory for everything that's going on in the cell, while the cytoplasm is where the real work happens. So let's get that messenger RNA out into our cytoplasm and let's get those proteins created. Another thing that you might want to make sure your students have a good understanding of is, do they understand that your table or my hula hoop represent the nucleus and that the rest of the class represents the cytoplasm where the work is taking place, and that, you know, genetic mistakes happen. And surprisingly, as many times as our DNA is replicating and as many times a day and in your lifetime that messenger RNA is sending that code out to make that protein, our body makes very few mistakes. And I think that's a miracle. So I thank you, and I hope you will check out more of our presentations on Kansas Corn STEM.